Our last speaker will be Mary Beth Wilhelm. Dr. Wilhelm got her bachelor's in Earth and Planetary Science from Cornell University, and then got a PhD in planetary at Georgia Tech. She was the youngest person hired at NASA in Earth and Planetary Science. Did I see that right? So she's super smart. She's also the PI of the Excalibur instrument, which does separation science. So, Mary Beth.
Leaving organic chemistry for the talk right before lunch. Thanks, Mike. So Mike asked me to put together a few slides about kind of an overview of biosignatures and kind of general techniques for life detection. So I'll preface this with a lot of the slides I'll show you has sort of my tilt to it. So a lot of stuff about Mars and a lot about ancient biomarkers, so mostly lipids and kerogen. But I tried to kind of bake in more of a high-level overview to get everyone sort of up to speed. So to first take a step back, think about why we want to do life detection missions. What are the kind of general motivations? So obviously the search for life remains a key NASA science goal. So that includes both extant and presently habitable environments, as well as ancient and sort of preserved remains, previously habitable environments. But I think it's sort of important to start thinking about this as more of a continuum. And there's going to be overlap in those two kind of distinct pools. Also, I think it's important to start to improve our understanding of early prebiotic chemistry and that sort of link to Earth. The very earliest epoch on Earth, when life developed, we don't have that record intact. So even if we encounter a planetary environment where life we don't fully get to biochemistry, I think there's some important data points there we can learn about what happens in an environment that doesn't quite make it all the way to life, and will help us to understand ourselves. Additionally, I think organic chemistry is cool. Understanding the carbon cycle is really interesting. I have friends that are magnetospheric physicists that would disagree with me, but I think that's also one of the byproducts of understanding organic or organic chemistry in these types of environments. So in the search for indicators of life, first you sort of start off with your basic research about life's origin, analog research, as Chris talked about. That's also important in the field of astrobiology, and I'm sure a lot of you folks do this as well. So we use that sort of body of knowledge then to inform the types of strategies we use, which targets we select, what sort of measurements are important, and how do you build out a complementary set of measurements, because no one measurement will be definitive. And then that then feeds into tech development. How do you collect your sample? How do you process it? What sort of analytical techniques? And then finally, all of that goes into then, choosing your destination and developing your mission concept. So what should we look for? It's likely that there are some aspects of life that are universal. That includes sort of the versatile chemical building blocks, complex multimeric biomolecules, containment structures, function-specific molecules. Arguably, all of those are required for life, and combined would be very compelling evidence of life. And so, starting to think about how then that feeds into something like a science traceability matrix, which is the holy grail of mission concept development. You have to be able to track from your basic measurement to the parameter you want to observe all the way through your analytical approach. So today I'm going to be talking a lot about sort of structural polymers, so lipids, and some of the techniques that kind of make sense for those specific molecules. But the examples I'm talking about are by no means comprehensive. So kind of where the field is at, there's been some work over the last couple of years by the Network for Life Detection to kind of go through the types of biosignatures we know and start to sort of categorize them on this sort of confidence scale. So what measurements can you make that help give you greater confidence in the definitiveness of a biosignature? And that goes kind of from the bottom of, is there carbon, all the way to the top, which is looking at distributions of identifiable molecular structures or components. But ideally, we want to make measurements across the spectrum, but kind of sort of prioritizing those measurements at the top because they can help give us more confidence in those measurements. So you hear a lot of folks in my field talk about biomarkers. This is one definition of biomarkers. So these are organic compounds that retain their biological heritage. But I think an important point is that that heritage is conserved in the molecular structure through geologic time. So when we talk about organic molecules, we also have to think about the sort of processes that will degrade those structures. So picking molecules that are already robust, I think is super important. It's also important to think about how these processes might work on other bodies. So in my fun cartoon, we have the example of a leaf which has chlorophyll in it. So the leaf falls. If you're lucky, that there will be a body fossil, there will be some sort of preservation of that leaf. But if you look into the rock and you pull out the biomarkers that are present, you can see the degraded structures from chlorophyll. So the chain breaks off from the ring, but essentially you can look at those molecules and understand about the metabolism of that original environment that's been dead for a really long time. So I think looking at molecular structures is a really powerful measurement that we can make, although by no means the only one. So it's also important to think about how these processes might work on other bodies, and that we don't make Earth problems necessarily problems that we're going to encounter on other planets, or there could be problems on other planets that don't occur here. So in designing life detection experiments, we need to be really careful to think through all of those different processes from synthesis all the way through degradation of organics. So on Earth, microbial processes mediate the majority of organic decomposition. Earth is an incredibly efficient recycler. Over 99.9% of material is recycled, which is pretty incredible. And the initial preservation conditions oftentimes dictate the long-term survival of biomarkers. It's also important to remember that most of what we know about biomarker preservation is from aquatic environments, so that's not always applicable to some other types of environments that we're looking at. So there's a whole number of things that the biomarker has to endure and survive before we eventually get to detection. Also, geologic deposits are not always stable. So this is an interesting figure that I found in a paper that was published about 30 years ago by Dave Demery and Jack Farmer, looking at the stability of different types of geologic deposits and in the context of what would be ideal for preservation. And obviously we've spent a lot of time looking at clays and shales and those sorts of things, and not that we're overlooking ices and halides and sulfates, but I think we're starting to realize that processes that might cause you to lose those deposits, like climactic warming or dissolution, may not be as much of a concern in places that aren't Earth. So I think, especially with sulfates, I know the recent Perseverance results came out and that we're finding organics and sulfates. I think it's important to start kind of looking at these, potentially less stable geologic deposits. Additionally, biomarkers themselves have a particular geologic longevity. So unfortunately, there seems to be this inverse correlation between the amount of information contained in your biomarker and the stability of it over geologic time, which is annoying. So DNA, in the best case scenario, is lasting a million or so years. And then you have amino acids, and that really kind of amino acid detection has been the focus of a lot of life detection over the last couple of decades because of the favoring of one of the enantiomers. But that enantiomeric excess on Earth only lasts about 10 million years. So you start to lose that information pretty quickly. And then proteins, humic complexes, these sorts of things can last about 100 million years. And so then finally you end up with lipids, which is what we see in really old deposits on Earth, at least the hydrocarbon cores. And I'm not going to get in a debate about how old the oldest lipid is, but it's on the scale of billions of years. So these are definitely geologically robust molecules. So then thinking about what is the majority of the organic content that we see in the rock record. So if you take your average rock, about 95% is minerals, the other 5% is organic matter. Of that organic matter, 85% is kerogen. And if you haven't heard of kerogen, it's not super surprising. This is a really complicated molecule. It's a large macromolecular mess, and the techniques you have to use to kind of break it into useful pieces are very difficult to implement. But I think it's pretty groovy. I've been trying to get more into looking at kerogens recently. So anyway, the other 15% is your soluble fraction, the so-called bitumen, and that's where you find your lipids hiding. So I've talked about biotic settings. It's important to note too that abiotic environments, you find the same types of organics in abiotic environments. So this pie chart is showing you the soluble fraction of organics in Murchison. And about 60% of that pie chart is made up of carboxylic acids, aromatic and aliphatic hydrocarbons. So the very same types of molecules that we're seeing kind of left over on Earth as well. So yeah, but it's important that we start to understand the distinction between biotic and abiotic organics. Because we want to be able to, no matter what sort of material we encounter, we want to be able to look at the features and patterns of that material to understand how it was synthesized. Yeah.
In a truly abiotic environment, would you expect maybe something a little bit more exotic than the things that we'll see in a biotic environment? Longer chain because there's nothing to consume.
Right. So in abiotic environments that we studied, so I'm showing one example here from carbonaceous chondrites. You actually see mostly shorter stuff, more simple things. And I have some graphs coming up that kind of show those separations. But yeah, like, you know, you made a good point that like we haven't studied every abiotic environment yet. And I think that is a big problem in thinking about how do you look for organics you don't know what they look like yet on a body we haven't yet explored. So we talked about that definition of biomarkers, about biomarkers retaining some sort of heritage in the molecules, and this is kind of what we're talking about. You have, you know, different types of lipids that undergo diagenesis, and you end up with what's called a molecular fossil that you can connect back to that original molecule. So it's important to note that like these features are maintained through the diagenesis processes and preserved in the rock record. And it's pretty powerful. Like you can study the remains of organisms that have been dead for 1 billion years. Like there's a lot of power in being able to look at these features and structures. So now kind of shifting into how do we do biosignature detection. And I want to step back to some of the early work that was done by Lovelock in the 60s. He was one of the guys that did some of the underlying sort of philosophy and thinking that went into informing some of the Viking experiments, and pointed out what I think has been sort of the basis for a lot of our kind of modern thinking on biosignature detection. So he showed that in carboxylic acids or fatty acids in meteorite settings have this sort of stochastic distribution, and then in biologic settings you have a maximum at a particular chain length that then has this other kind of funny pattern that's driven by enzymatic production. So I don't think we've moved too far past this yet. I think we're finally the last decade, it seems like the field's sort of finally starting to move forward. And I think, yeah, Lovelock's actually a really interesting guy. I looked him up last week. He actually passed away about a month ago. He was 103. And he also worked for MI5. But anyway, totally worth going down the Wikipedia rabbit hole on him. So, but kind of back to that point about we're seeing these patterns, what do the patterns mean? The molecular form is driven by the biologic function. So in the case of these membranes, the chain lengths, the types of modifications you see to the membrane lipids, it has to do with enabling the proper membrane geometry. In the talk earlier, you're talking about resistance to different types of physicochemical parameters. So these things aren't random. They're coming from a specific driver, and there's lots of textbooks I can point you to if you want to start to understand why those features are there. This is a cute little graphic that Mike gave me that I really like that kind of shows one example of this with in the case of terrestrial cell membranes. So we have two major types. We have bilayers and monolayers, one for bacteria and eukaryotes and the other for archaea. But essentially when you start to look at the molecules, their length, their function, they're the same. And that the size is driven by, if it's too small, it's going to diffuse away, and if it's too big, it's not going to have the right features. So again, kind of just driving that point home that these molecules are made for a reason. So something that I did with my team, and one of my students in particular led as our COVID project, was to go through the literature and review about 1,500 samples and start to pull out of the published literature and quantify different parameters that we thought would be origin diagnostic. So looking at these features and distributions that tell us about how the molecule is made. So that not only includes chain length, as Lovelock pointed out, but also where the position and the number of double bonds are in chains, where branch points are located, how many they are, how long they are. And we've been finding some really interesting results. Nothing too surprising, but sort of now we have quantified the kind of bounds on what we're looking for. So it shouldn't be too surprising that in the terrestrial systems, you have these longer, so this is minimum fatty acid chain length versus maximum. You sort of have these longer chains. And then in the meteorites that have been studied, they're shorter. And then the most abundant one is different. In terrestrial systems, you have C16 dominating, whereas in meteorites, you have C2. So what's cool about this data is now we have this huge data set that we've been exploiting, and Denise has done a great job wrangling this data, and we'll be publishing it hopefully, hopefully this month. But the molecular features are additive in terms of helping us understand origin. And so when you start to add up all those diagnostic features and patterns that we quantified, and you plug them into a principal component analysis, suddenly you see this big separation. And I think that separation is going to be really important in thinking about exploration of an unknown body, because we don't yet know what that's going to look like. So where I sort of hope this field goes is to not just look at life detection as a binary of this is biologically driven, this is abiotic, but we have to recognize that we're going to encounter multiple types of abiotic synthesis. We could encounter known or unknown biological synthesis mechanisms, and then all of those organics are going to be degraded. So they're going to be subjected to radiation, oxidation processes, a lot of geological reprocessing. So when we start to make the technology and the instruments to look for these materials, we have to make sure we encompass all of the potential modifications that have been made to those structures. And one of the first things that at least I think we're starting to get there with Mars is starting to draw out really where that abiotic baseline is, because we really don't quite know that yet for something that's not a meteorite, or at least we don't know it very well. So this is a slide I stole from Ali Fox, which I think kind of sums it up well for Mars. So you have all these types of potential organic synthesis processes, so delivery from meteorites, geochemical reactions, radiolytic processes, and igneous processes, and the holy grail, of course, which is life, that have all gone through deposition and then different types of potential degradation pools to eventually decompose. But in some cases, through preservation mechanisms, you end up with what we've detected, which is the current Martian organic inventory. So some additional considerations that I just wanted to point out that, as keeping in mind as we start to think about potential concepts to move forward with. Inorganics are really difficult and can interfere with analysis. So that includes oxidants, salts, and sulfurous minerals. Biomarkers are also, I've yet to see an environment on Earth where biomarkers are distributed homogeneously. So we're going to have to deal with some sort of heterogeneity issues in developing life detection instruments and missions. Also, organic material is complex. It's been sitting around for a long time, and Heather was like, make sure you talk about this point, that organics have complex molecular interactions with minerals. And so they can be bound inside, they can be attached on the outside. There's a really good review paper, if you are interested, by Keel and Mayer that was published 15 years, or is that 10 years ago, about this, if you're interested in that. But those are non-trivial things that we're going to have to overcome in order to make that final detection. And also, being able to achieve a very low limit of detection, I think, will be critical. It's not good enough just to detect organics. We're going to really get at those fine features in the molecules. We're going to need to see things that are very low in abundance. And so you can see, at least for the case of Mars, we're sort of getting at lower potential limits of detection, and hopefully with MSR, with return samples, that number should go down quite a bit. But it's also important to note you can hit this problem on two sides, both on the front-end sample processing and then the back-end analytical limit of detection. So there's a couple ways we can try to solve that problem. Additionally, the quality of molecular information is really critical. Again, going back to our sort of Mars case, previous Mars missions have so in the case of Viking and Curiosity, have used pyrolysis techniques to liberate organics. And so those, while successful in getting out some of the smaller components of larger molecules, it's really hard to tell what larger structures those smaller molecules are coming out of. And there's a couple of reasons for that being so difficult. When you're heating up complex geologic material with organic material to high temperature, the chemistry gets really complicated really fast. And then with Perseverance, we are now kind of getting these broad indicators of organics. But again, it's hard to kind of look at these structures or indicated structures and get back to origin. How is this stuff made? Where is it coming from? And so that's kind of my hope for Mars moving forward. So on Earth, there's really well-defined, widely used analytical approaches to biomarker analysis. They're efficient, they overcome all those issues with the minerals, and they maintain the organics, structural integrity throughout that, so when it's supplied to your ultimate analytical instrument like a mass spec, you have the whole thing going in. It's been demonstrated on hundreds of thousands, if not more, terrestrial samples. However, it has many steps, employs large volumes of solvents and consumables. As Chris said, sample processing is really hard, and I think it's going to be one of the big challenges to doing it well. So thinking about how we might apply that to maybe an icy moon sample, I think is going to be really difficult because of all of the potentially interfering inorganics that will be there and the low signals. So that, I think, is a pretty hard problem. So thinking forward about the technology that would have to be integrated with, I think we're going to want some sort of processing system, maybe in the liquid phase, that can be integrated with chemical sensors, spectrometers, different types of microscopy. There's a lot of technology lineage and heritage already out there that we can exploit that's maybe not always tapped in the planetary science world, such as the CubeSat world and space biology world. And also in human health and performance. I have a bunch of friends at Ames who have a bunch of sensors they use for astronaut health, and I think there's some technology there that we might be able to pull into our thinking. And there's a lot of emerging applications. I'm really excited about the work going on with CLIPS. I think we're going to have a lot of more mature technology really quickly. So I'm excited to see where this field goes in the next 10 to 20 years. And so from my last slide, I just sort of have my initial brainstorm of kind of all the trade space of types of instruments out there, architectures, potential measurements. And so with that, I'll leave this up and take any questions.